I am going to do a quick run through of some of the science of Parkinson's disease. We've already had a wonderful talk on exercise. The topics that I chose are not necessarily the ones that are most prominent on the minds of the scientists studying this, but they're absolutely on the minds of the people that I see in the clinic, and this talk is driven primarily by what I'm asked about. And I will take a little bit of liberty at certain parts of it, filling things in that may not be 100% proven or 100% backed by science, but most of what I'm going to talk about is pretty solid and does put together an interesting story that is the basis for some of the clinical trials being done now. So these are the topics, and I'll be going through them sequentially. So sorry to start your Saturday with biochemistry, but there's a requirement to understand this part of biology to understand the rest of the talk. Genes produce proteins. Proteins then fold into shapes. When they fold correctly into shapes, they interact with one another to perform a function. And that's really how things run. So in this particular case, kind of a saddle and rider type of setup to allow the two proteins to come together to function. When a protein misfolds for a variety of reasons, it will not form the same three-dimensional shape that it did before, and that can lead to lack of interaction with other proteins and a lack of function. Misfolded proteins can also do bad things. They might start to interact with other proteins or cause other functions that end up being toxic to the cells. In the case of Parkinson's disease, misfolded alpha-synuclein is toxic to the cells. That has been proven over the decades. So if we look at a pathological process, so if there's a mutation in this alpha-synuclein protein, if there's cellular stress, if there are a variety of disease processes, which I'll talk about briefly, that can transform normally folded, normally functional alpha-synuclein into an abnormally folded, abnormally acting protein. There's been a lot of talk over the last year or two with paraquat lawsuits that have come out, people exposed to paraquat having Parkinson's disease. The reality behind this is that this is epidemiological data that we've probably known for 30 years, that herbicides and pesticides, chronic exposure, there's a higher risk for developing Parkinson's disease. So it's gotten attention recently in terms of lawsuit purposes, but we've known this for a long time, and a lot of biochemical and cell biological animal models are based on this information. The reason I have a question mark at the end is that despite the correlation with epidemiology, it's still not 100% proven that exposure to these agents causes Parkinson's disease proper that people experience. Now, my suspicion, I think the bulk of people on the professional side of this think that there's a combination of chronic exposure to these agents and kind of a genetic susceptibility or inability to protect the brain against them that really does cause these disorders. But it's not a proven one-to-one entity. These abnormally folded alpha-synuclein molecules get kind of sticky, and they stick together, and they form initially what are called protofibrils, so kind of lines of the protein together, stuck together. And what do you know but mutations in alpha-synuclein itself, cellular stress, disease processes otherwise, and pesticides and herbicides have all been shown to variable degrees to promote this stickiness and promote this fibrillization, this abnormal fibrillization of the protein. These protofibrils will then start to aggregate together into bigger and bigger clumps. It's called aggregation. And the ultimate visible aggregation of these proteins is what we call the Lewy body. Dr. Lewy, back 100 years or so ago, noticed these things, and then he eventually got his name on the inclusion bodies themselves. We really don't know if the Lewy body itself is pathological or not. The cells that we look at are the cells that are still alive when an autopsy is done and the tissue is looked at. And many of us think that maybe the Lewy body is the way that the smart cell is taking up all this trash and trying to stick it into the closet so it can still function. However, these Lewy bodies and the pathology does tend to occur most prominently in cells that use dopamine to help us to move big, move fast, not have tremor, and have normal muscle tone so that we can kind of move and do our thing. And ultimately, these cells do start having trouble with function, and then they start to die. And when enough of these dopamine cells die, then the clinical expression of Parkinson's disease occurs. So what is known, probably for 2 decades now, is this protofibril level intermediate in the process of aggregation is very toxic to cells in a variety of ways. Some information, but I think less clear whether the, kind of what is called the monomer, the single cell, the single molecule is really toxic, and whether when these proteins aggregate further, if that is still as toxic as the protofibril. This process is called seeding by some, so it's like a crystallization almost, and seeding is we'll talk about that in a moment in terms of some of the new exciting information that came out of the Michael J. Fox Foundation funded research recently. So the seeds, so to speak, have been proven over the last decade and a half to actually spread from cell to cell. So from an unhealthy cell to a healthy cell to cause problems. And Heiko Brock, now going back almost 30 years, which I can't believe, but going back about 30 years, did show that in pathological, in brain section samples taken from all comers who had passed away, that it does look like there is a progression of Parkinson's disease from the base of the brain stem to a little bit farther up in the brain stem and then throughout the brain. This area here, kind of the top of the brainstem, base of the brain, contains the dopamine cells that have difficulty. So this area of the brain is for olfaction or smell, and a lot of people lose or have reduced smell. This area of the brain at the bottom, so this is the smell area here. You can all see my tremor, and then the lower areas here, some of the autonomic or automatic functions. Once the pathology hits kind of the mid-range here, that's when tremor, stiffness, and slowness develop. So that there's a fair amount of evidence that this pattern of progression occurs as these alpha-synuclein seeds spread. There's also more recent evidence that these seeds may spread from the gut or the intestines to the brain. And there is strong evidence from some people that these abnormally folded and abnormally seeded alpha-synuclein aggregates are found in the intrinsic nervous system of the intestines, connected to the brain by the vagus nerve, and guess what, connected to the lower part of the brain where or the brainstem where disease may start. This process is known to occur to cause prion disease. So the mad cow disease from 80s, 90s, whenever that was, when people in England were eating the tainted beef and then having degenerative disease of the brain, that's exactly what happened. They ingested these proteins which spread from the gut to the brain and then ravaged the brain. So in terms of human pathology, it is a mechanism that is known to happen. And so then the question is this really what happens in Parkinson's disease? Well, there's lots of kind of each level evidence, but there are there may be people where the PD starts in the gut and moves to the brain, maybe starts in the brain, moves to the brain. This is really not fully developed, but it is the source of a lot of research and interest these days. And whether people know it or not, this is the source of the question of, should I use probiotics? So in other words, if I improve the health of my gut, not only will that help to absorb medication better, et cetera, but if I improve the health of my gut, will that reduce the spread of this alpha-synuclein to the brain and then through the brain? And so the quick answer to this question is nobody really knows yet. What probiotics do you use? How do you... We just don't know the specific answer, but this is the reason why people are asking about it and why people talk about the gut-brain interaction. So the media, Michael J. Fox Foundation, has just spread the word about this. It's alpha-synuclein seed amplification assay, which is receiving a lot of press and interest because these seeds can actually be detected in spinal fluid. So you get a lumbar puncture, take out some spinal fluid, these can be detected. You should know that these seeds can also be detected in the gut by a gut biopsy, which we don't really do, or the skin by a skin biopsy. There's actually a commercially available test for the same thing. It's just that the sensitivity of this spinal fluid assay is much higher, and in a number of people in this study that was just published who did not have Parkinson's disease but did have some prodromal symptoms of maybe acting out dreams or constipation, escalated anxiety, they had a positive test for these proteins and then later developed Parkinson's. So the excitement behind this is, and particularly if this could be a blood test, which is a lot easier than getting spinal fluid, could this be added by your primary care doctor to your CBC and your chemistry panel and your thyroid hormone, your Parkinson's test? Whoops, you've got alpha-synuclein seeds. And then when we have, we'll get you exercising, which we already know helps, but when we have disease-modifying curative therapies, maybe based on tests like this, we can actually start them before the tremor, stiffness, slowness develops and just kind of flatten the curve there. So that's where the excitement comes from that, and that's a major advance. How does the brain itself fight against the alpha-synuclein seeding and spreading? The brain's pretty smart, and it's battling these things along with you as they're developing. So I told you that these components of the pathway, and particularly the protofibrils, are toxic, but the cells have some defense mechanisms. One is called a proteasome, and basically once these proteins abnormally fold, they're tagged and then dragged to the proteasomes where they basically shred up the protein. We also have lysosomes, which are basically bags of acid. And again, when the abnormal proteins are produced, they're tagged and dragged to the lysosome where they're digested. Okay, so we shred and digest. Pretty nice system to try to battle down proteins that are abnormally folded or modified in some way. Well, if this is happening, how do we still get Parkinson's? It's kind of a balancing act between the production of the problem and the fighting of the problem. And so there are a number of mutations in genes that are known to cause Parkinson's disease, and there are a much higher number of mutations or polymorphisms that increase the risk for Parkinson's but don't cause it. So probably that genetic susceptibility versus environment dynamic. But these 7 genes that are listed here, when mutated, can influence the production of these protofibrils and fibrils to build them up. These are the genes that are studied in the Parkinson Foundation PD generation study that you can get done for free. These are the most common forms of genetic Parkinson's. So we can actually test for these, And then as more is learned about the mechanism of each of these genes, if you have a GBA mutation, for example, there may come a drug that battles that down. So if we have that information from the PD generation study, we may be able to put you on that modifying therapy. These mutations also, for the bulk of these genes, will actually reduce the effectiveness of these shredders and acid pots, the proteasomes and the lysosomes. So they kind of battle down, they build up the toxic influences and battle down the fight against them. And then a variety of mechanisms that scientists are studying, including the pesticide herbicide exposure, oxidative stress inflammation, and a variety of other mechanisms will not only promote protofibril production and the aggregation of the alpha-synuclein, but also reduce the capacity of the cell to battle this back. So with this framework in mind, scientists are working on ways to attack each stage of the process. There are compounds, buntanetap is actually being studied at a variety of places, including Ohio State, to try to reduce the production of alpha-synuclein to begin with, to reduce the process. And actually in recent years, albuterol, which is the inhaler compound, seems to reduce production of alpha-synuclein. So who knows where that might go. There are some compounds that have been shown to reduce the aggregation process itself that are in clinical trials at various levels, and tricyclic antidepressant medications that I do use in the clinic in the test tube can work for this. We don't know how that really affects Parkinson's, but there are compounds being studied for that part of the process. There are neurotrophic factors that protect cells in a variety of ways. So if you have a cell that's having some trouble, these factors can help to reduce the risk of dysfunction and death. And GDNF is something that we're actually working with a company to hopefully bring into clinical trials to Ohio Health in the near future. There are a variety of antibody approaches that will neutralize these abnormally folded proteins, again in clinical trials at various levels, including immunization type studies. And those of you who have seen the news recently over the last few months for anti-Alzheimer's drugs, the three that have been introduced recently are all antibody approaches that go for the abnormally folded aggregated beta amyloid protein in Alzheimer's disease to try to neutralize it and bring it down. So this mechanism is already at various levels of FDA approval for Alzheimer's disease. So hopefully it will follow for Parkinson's in the near future. These are Daniel gave me a picture of his arms to use for my talk. There are a few compounds and small molecules that will actually beef up the the proteasome shredding and the lysosome acid breakdown of these proteins. So a variety of levels of clinical trials to try to battle down the process of Parkinson's disease. I have 1 minute and 11 seconds left, which is about all I need. The Parkinson's Glove I'm asked about at least once a day, so I thought I would say a little bit about it. And it was fun to learn about this in terms of the history. This mechanism actually goes back to the 1900s, where neurologists at the time would talk with people who had tremors, Parkinson's disease before it was named Parkinson's disease. Folks will say, you know, I had a really long bumpy ride in the carriage and my tremor wasn't as bad for a while and I could walk better and I wasn't as slow as stiff. What's going on? And so people have actually done studies of putting people on chairs and jostling them around and shaking them back and forth to see what happens. And that hasn't worked out too well, so we don't have a shaker chair yet. Now, when my wife's shaking me, maybe that helps, but we don't have that kind of therapeutic in our office quite yet. But what scientists have learned is that, and this makes some sense, if this is a representation of 3 different cells in the brain, and each cell is kind of burping along in its own pattern. It's receiving signals, it's sending the next signal, but it's doing so in its own way. In Parkinson's disease, there's evidence of synchronization. So basically, instead of the cells kind of doing their own thing, functioning their own way, they start to function in synchrony together, and that can correlate quite a bit with aspects of the walking issues, aspects of the slowness of movements, aspects of the stiffness, and maybe less so the tremor. But what some very, very clever people, and Dr. Tass, who is the one doing the Parkinson's Glove, has been instrumental in this. Folks have shown that vibration will actually desynchronize the cell firing. So basically take their firing pattern back to a more normal state, and that correlates with better walking, to some degree less tremor, but less stiffness, less slowness. And so that's the idea behind the glove, to provide that type of vibration at fingertips. And interestingly, levodopa, dopamine replacement therapy, will do this to some degree. And guess what? It's one of the thoughts behind the mechanism of deep brain stimulation, to actually go in to the problem area and electrically desynchronize those cells. And so the technique itself is called Vibro Tactile, so it's vibrating on touch surface, coordinated reset stimulation. They describe it as it's like the vibratory buzz of a cat purring, about that level of sensation on the fingertips. And the studies collectively have shown that 10 out of 11 people that they did this in developed improvements, like Dr. Korkos was showing, in gait, tightness of muscle tone, and slowness of movement, more so than tremor. So proof of principle that doing it here rather than shaking you on a chair can be helpful. And the status now is that bigger clinical trials are being planned. Where do we stimulate? How do we stimulate? Etc. So it's a very, very interesting topic, and I'm glad people asked me about it because I hadn't really studied into it to this degree. So believe me, and you just saw one of them, but scientists are really working on this really, really hard, trying to figure this out. Please do support them and stay tuned. Every year there's more and more good news towards help for this condition. All right, so I'm over time, but you know, I took it. So thank you.